So, good evening, guys. Today is the last session. This is an extra class, which will be of 1 hour. So, this session, we'll also discuss some of the sample assignment questions from last year of this course, Biochemistry IIT Madras course, taken by course instructor Professor K. Subramaniam, and we'll be discussing some of the last year questions, and accordingly we'll try to keep the session as interactive as possible, and we'll not only discuss the questions and the solutions, we'll also try to discuss the concepts behind those questions so that it's easier for us to solve similar type of other questions. So without further ado, we'll move on to the first question for today. The TCA cycle or the Krebs cycle, TCA cycle is the same as Krebs cycle. So it consists of many steps which take acetyl-CoA through many intermediates, extracting electrons along the way by reducing electron carriers like NAD and FAD. Which of the following enzymes mediate steps that reduce NAD FAD? Okay, so these are the four options that we have today. Citrate for this question: citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase complex, and succinyl CoA synthetase. So we'll stop for 2-3 minutes, after which we'll come back to discussing the question. So in any case, if you want to try the question, the field is open. Yeah. Anybody? Okay. So the answer for this question will be isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase complex. So first talk about Krebs cycle, TCA. So acetyl-CoA is a substrate of this cycle. It rises from the catabolism of many carbohydrates, lipids and amino acids, and it is an energy-rich molecule composed of coenzyme A and acetic acid joined by a high-energy thiol-ester bond. Right. So after glycolysis, we can move to the citric acid cycle or the Krebs cycle. So this is the entire story of the Krebs cycle. If you, of the Krebs cycle, glycolysis, which gives rise to pyruvate, and then it under aerobic conditions, it will go forward for the Krebs cycle, or else if there is fermentation happening under anaerobic conditions, it might get converted to it can undergo fermentation basically. Otherwise, we'll go ahead to the acetyl-CoA production, which is a 2-carbon compound. From there, we start the Krebs cycle. Citrate is 6-carbon compound, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. And obviously, we have a bunch of enzymes that catalyze this process in getting involved in the cycle. So TCA cycle generates 2 CO2s, 3 NADHs, 1 FADH2 and 1 GTP for each acetyl-CoA molecule oxidized. And the cycle enzymes are widely distributed among. So the complete cycle is functional in many aerobic bacteria. The facultative anaerobes do not use the full TCA cycle under anaerobic conditions, and this cycle is an important source of energy and provide carbon skeleton for use in biosynthesis. So only 4 ATP molecules is synthesized directly when one glucose molecule is oxidized to 6 carbon dioxide molecules by glycolysis and TCA cycle. So most ATP generated comes from the oxidation of NADH and FADH2 in the ETC, the electron transport chain. Now what's the ETC, the electron transport chain? So the mitochondrial electron transport chain is composed of a series of electron carriers that operate together to transfer electrons from donors like NADH and FADH2 to acceptors such as O2. So the electrons flow from carriers with more negative reduction potentials to those with more positive potentials and combine with O2 and H plus to form water. So the electron transport chain carriers located in the inner membrane of the mitochondria or in the bacterial plasma membrane. So the process by which energy from electron transport chain transport is used to make ATP is called oxidative phosphorylation. Right? So what is the ATP yield from the entire aerobic oxidation of glucose? First we go through the glycolytic path. We have discussed this in one of the previous classes. So for substrate level phosphorylation, there's 2 ATP generated. For oxidative phosphorylation with 2 NADH, there's 4 to 6 ATP. Now from 2 pyruvate to 2 acetyl-CoA, there's 6 ATP oxidative phosphorylation with 2 NADH. And then the TCA tricarboxylic acid cycle or the citric acid cycle or the Krebs cycle, there is 2 ATP generated from substrate level phosphorylation, and then oxidative phosphorylation with 6 NADH will give rise to 18 ATP. Oxidative phosphorylation with 2 FADH2 will give rise to 4 ATP. So total aerobic oxidation of glucose yield will be per ATP will be 36 to 38 ATP. Now, aerobic respiration is much more effective than anaerobic process that not involving electron transport and oxidative phosphorylation. Okay. So many organisms, when they move from anaerobic conditions to aerobic conditions, they'll reduce their rate of sugar catabolism and switch to aerobic respiration. And this phenomenon is known as the Pasteur effect, right? This is advantage to the organism is less sugar must be degraded. Correct, less sugar must be degraded to obtain the same amount of ATP when the more efficient aerobic process can be employed. Okay, so the partial effect basically describes the available oxygen inhibits ethanol fermentation. So driving yeast to switch towards aerobic respiration for increased generation of the energy carrier, adenosine triphosphate or ATP. Okay, so it basically refers to how the cellular presence of oxygen causes in cells a decrease in the rate of glycolysis and also a suppression of lactate accumulation, and the effect occurs in animal tissues as well as in microorganisms, right, belonging to the fungal kingdom. So this effect was described by Louis Pasteur in 1857 in experiments showing that aeration of yeasted broth causes cell growth to increase while the fermentation rate decreases based on lowered ethanol production. So, like yeast, fungi being facultative anaerobes can either produce energy through ethanol fermentation or aerobic respiration. So, when the oxygen concentration is low, the two pyruvate molecules formed through glycolysis are each fermented into ethanol and carbon dioxide, right? So while only 2 ATP are produced per glucose, this method is utilized under anaerobic conditions because it oxidizes the electron shuttle NADH into NAD plus for another round of glycolysis ethanol fermentation. So if the concentration of oxygen increases, pyruvate is instead converted to acetyl-CoA used in the citric acid cycle and undergoes oxidative phosphorylation. So per glucose, 10 NADH and 2 FADH2 are produced in cellular respiration for a significant amount of proton pumping to produce a proton gradient utilized by ATP synthase. So while the exact ATP output ranges based on considerations like the overall electrochemical gradient, aerobic respiration produces far more ATP than the anaerobic process of ethanol fermentation. So the increased ATP and citrate from aerobic respiration allosterically inhibits the glycolysis enzyme phosphofructokinase 1 because less pyruvate is needed to produce the same amount of ATP. Okay. So despite this energetic incentive, people have shown that these continue to partially ferment available glucose into ethanol for many reasons. First, glucose metabolism is faster through ethanol fermentation because it involves fewer enzymes and limits all reactions to the cytoplasm. Second, ethanol has bactericidal activity by causing damage to the cell membrane and protein denaturing, allowing yeast fungus to outcompete environmental bacteria for resources. And third, partial fermentation may be a defense mechanism against environmental competitors, depleting all oxygen faster than the yeast regulatory systems could fully switch from aerobic respiration to ethanol fermentation. Okay. So if we talk about anaerobic respiration, many bacteria have ETC electron transport chains that can operate with exogenous electron acceptors other than O2. Okay. The major electron acceptors are nitrate, sulfate, and carbon dioxide, but metals and a few organic molecules can also be reduced. Now, some bacteria can use nitrate as the electron acceptor at the end of the electron transport chain and still produce ATP. This process is called dissimilatory nitrate reduction, and nitrate may be reduced to nitrite by nitrate reductase. Okay. However, this is not effective way of making ATP because the large amount of nitrate is required for growth. Nitrate molecule will accept only 2 electrons. The nitrate formed is also toxic. So therefore nitrate is further reduced to nitrogen gas. This process is known as denitrification. Okay, and denitrification is carried out by some bacteria like Paracoccus denitrificans, Pseudomonas, and Bacillus. Okay. Now some bacteria like methanogens use carbon dioxide or carbonate as a terminal electron acceptor. They reduce CO2 to methane. Sulfate can also act as the final acceptor in bacteria such as disulfovibrio. It is reduced to sulfide and 8 electrons are accepted. If we move to lipid catabolism, microorganisms use lipids as energy sources. So triglycerides or triacylglycerols, esters of glycerol and fatty acids are common energy sources. So they can be hydrolyzed to glycerol and fatty acids by microbial lipases. The glycerol is then phosphorylated, oxidized to dihydroxyacetone phosphate and catabolized in the glycolytic pathway. Now, fatty acids from triacylglycerols and other lipids are oxidized in the beta oxidation pathway. After conversion to coenzyme A esters, fatty acids are degraded to acetyl CoA, which can be fed into the TCA cycle or used in biosynthesis. The cycle also produces acetyl CoA, NADH, and FADH2. So NADH and FADH2 can be oxidized by the electron transport chain to provide more ATP. And lipid fatty acids are a rich source of energy for microbial growth. Now, talking about protein and amino acid catabolism, some pathogenic bacteria, fungi, food spoilage, and soil microorganisms can use proteins as their source of carbon and energy. So they secrete protease enzymes that hydrolyze proteins and polypeptides to amino acids, which are transported into the cell and then catabolized. So the first step will be deamination in amino acid use, removal of the amino group from an amino acid and transferred from an amino group to an alpha keto acid acceptor. The organic acid resulting from deamination can be converted to pyruvate acetyl CoA or TCA cycle intermediate and oxidized in the TCA cycle to release energy. It is also can be used as a source of carbon for the synthesis of cell components. Excess nitrogen from deamination may be excreted as ammonium ion, thus making the medium alkaline. Right? Now coming to citric acid cycle or tricarboxylic acid cycle, Krebs cycle. So it was given by Sir Hans Adolf Krebs. He was a German-born British physician and biochemist, pioneer scientist in study of cellular respiration, best known for his discoveries of two important chemical reactions in the body, urea cycle and citric acid cycle. Nobel Prize in Physiology and Medicine 1953. And with Hans Korbach, he also discovered the glycoxylate cycle found in plants, bacteria, protists, and fungi. Right? So, if we have to show the outline of the Krebs cycle, we have glucose 6-carbon glucose, which gets converted to pyruvate. We have 3-carbon 2 pyruvate, right? Then which gets converted to acetyl CoA, 2-carbon. Then oxaloacetate is added to it, which is 4-carbon compound, and he ultimately gives rise to citric acid. And then CO2 is given out, and again the chain cycle continues. Oxaloacetate is formed. So this is how it happens. If you see, this acetyl CoA, oxaloacetate joins, and then citrate is formed, and then from the citric acid cycle again it comes back to oxaloacetate. Okay, so the compound, so everything in the reactants and products have been shown here. The compounds of the cycle have been shown in red and the enzymes have been shown in blue. Okay, so be it carbohydrate, protein or fat, after digestion and absorption, carbohydrate gets converted to simple sugars, mainly glucose. Protein gets converted to amino acids and fat gets converted to fatty acids and glycerol. And then the catabolism process starts with acetyl CoA and then citric acid cycle. Okay. So if you can see, from amino acids there will be transamination. Correct. So these are the basic amino nitrogen glutamate can be transformed to NH3 by deamination. Acetyl CoA will move forward with the citric acid cycle, but this will move forward with the urea. So this is how everything happens. This is urea. This is basically liver, protein, amino acids, glucose, glycogen. Then you see the hepatic portal vein. Then you see the RBCs. Okay, then you see what happens in the muscles, lactate and everything, erythrocytes. Excuse me. So this is the cytosol. This is a ribosome. Protein is being made. This is the mitochondria. Okay. So glycolysis happens in the cytosol, and then inside the mitochondria, the Krebs cycle takes place. Okay. So you can see Krebs cycle is inside the mitochondrial membrane, and inside the mitochondria is the Krebs cycle, citric acid cycle. So these are the players of the game. Acetyl coenzyme A, acetyl CoA, the structure of acetyl coadenosine triphosphate, then acetyl group over here. Okay, pantothenic acid residue and beta mercaptoethylamine residue. Now we know about structures of adenine guanine, which are basically purine nucleotides. This is structure of glucose. This is structure of pyruvate. Okay. So this is adenosine triphosphate. Correct. ATP. This is flavin adenine dinucleotide. Just to show you a schematic of all the structures. It's a nicotinamide adenine dinucleotide NAT. So these are the enzymes of the citric acid cycle. Citrate synthase converts, adds acetyl-CoA and oxaloacetate basically together to form citrate. Then aconitase basically converts citrate to cis-aconitate. Then aconitase again converts cis-aconitate to isocitrate. Then isocitrate is converted to oxalosuccinate by the help of isocitrate dehydrogenase. Then oxalosuccinate gets converted to alpha-ketoglutarate by the help of isocitrate dehydrogenase. Then alpha-ketoglutarate gets converted to succinyl-CoA with the help of alpha-ketoglutarate dehydrogenase complex, which further gets converted to which is basically succinyl-CoA. Succinyl-CoA gets converted to succinate using succinate thiokinase, which gets converted, succinate gets converted to fumarate by the use of succinate dehydrogenase, then fumarate to malate by the use of fumarase, and then malate to again oxaloacetate by the use of malate dehydrogenase. You can see it's a very complex reaction. Okay, so this is basically the schematic of the Krebs cycle. Now, what do we mean by anaplerotic intermediates and cataplerotic intermediates? We have another question regarding this topic, so we'll discuss this terminology in the next question. But for now, if we have to see the entire schematic of the Krebs cycle, this is what the entire schematic entails. So the nucleoside diphosphate kinase catalyzes the exchange of terminal phosphate between different nucleoside diphosphates and triphosphates in a reversible manner to produce nucleoside triphosphates. Okay, so the citric acid cycle, that is a common mode of oxidative metabolism in most organisms, is mediated by 8 enzymes that collectively convert one acetyl-CoA to 2 carbon dioxide molecules so as to yield 3 NADHs, 1 FADH2 and 1 GTP or ATP. The NADH and FADH2 are oxidized by O2 in the electron transport chain with the concomitant synthesis of around 11 more ATPs, yielding about 12 ATPs for one turn of citric acid cycle. Now, several anabolic pathways cataplerotically utilize citric acid cycle intermediates as starting materials, and these essential substances are replaced by anaplerotic reactions of which the major one is synthesis of oxaloacetate from pyruvate and carbon dioxide by pyruvate carboxylase. So the enzymes of citric acid cycle act as a functional unit that keeps pace with the metabolic demands of the cell. So the flux controlling enzymes appear to be citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, and the activities are controlled by substrate availability, product inhibition, inhibition by cycle intermediates, and activation by calcium. Okay. Now, oxidation of fatty acids, activation, transport, and oxidation. So, you see, this is how it happens. This is outer mitochondrial membrane, inner mitochondrial membrane. Okay, so it gets basically carnitine acyltransferase 1 helps in the transport. This carnitine acyltransferase 2 inside in the matrix region. Okay, and this is acyl carnitine translocase in the inner mitochondrial membrane. So activation, this is the process of activation, if you can see. Then transport happens basically, and then oxidation happens inside. Correct? So this is it's an entire schematic of activation. Adenosine, then acyl-CoA and AMP, ATP, fatty acyl adenylate, fatty acyl-CoA. Okay. Now transport, quaternary ammonium compound involved in metabolism in most mammals, plants, and some bacteria as well. So they may exist in two isomers, D-carnitine or L-carnitine, as well optically active, and at room temperature it's a white powder, water soluble deuterion with low toxicity. Now in animals, as the L-enantiomer only and D-carnitine is apparently toxic. It inhibits the L-carnitine, and it is accumulated in cardiac and skeletal muscles as it accounts for 0.1% of its dry matter. It is synthesized from lysine side chains to keep up with the needs of energy production, and some individuals with genetic or medical disorders like preterm infants cannot really make enough. So this makes carnitine a conditionally essential nutrient for them. Okay, so you can see the transport. Cell transferase 1 is CAT1, and carnitine translocase is also there. And then we have carnitine acyltransferase 2. Okay, so this is the entire schematic of transport. Okay. So basically what happens is that the two enzymes carnitine acyltransferase 1 and carnitine acyltransferase 2 comprise a cycle that delivers a fatty acid as an acyl carnitine derivative, right? Okay. So if you see, this is the TCA cycle shown as a part of the central pathways of energy metabolism. If you see from this, It looks so small, but still it's so complex. You can imagine how complex the entire scenario is. So you can see the three stages of metabolism. And there are polymers, and they get converted to monomeric building blocks and activated acetate. Then these are the end products. So the catabolic pathways in stages 1 and 2 converge on stage 3, the Krebs cycle, and the Krebs cycle is a final common pathway of metabolism, right? Protein will get converted to amino acids, complex carbohydrate to glucose and other sugars, and fatty acids and glycerol from fat, correct? So this is an overview of the Krebs cycle. So you have already talked about all this, how it accounts for majority of the carbohydrate, fatty acid and amino acid oxidation. It also accounts for majority of the generation of these compounds and others as well. So it's amphibolic because it can act both catabolically and anabolically. Okay, so you can see who are the scientists who showed these kind of reactions. So Martis and Moore showed that pyruvate and oxaloacetate could form citrate non-enzymatically by the addition of peroxide under basic conditions, whereas Krebs showed that succinate is formed from fumarate, malate or oxaloacetate, and this is interesting since it was shown that the other way worked as well. So the interconversion rates of the intermediates was fast enough to support respiration rates. Substrate basically will have to flow across the outer and inner parts of the mitochondria. So Nathan Kaplan and Fritz Lipman discovered coenzyme A and CoA and line and showed that acetyl-CoA was intermediate from pyruvate to citrate. So it was a joint effort of a lot of scientists to come up with this discovery. So acetyl-CoA is a high energy compound. Delta G for the hydrolysis of its thioester is minus 31.5 kilojoules per mole, making it greater than the hydrolysis of ATP. So, and pyruvate dehydrogenase converts pyruvate to acetyl-CoA and CO2. Okay, so pyruvate dehydrogenase is a multi-enzyme complex. There are groups of non-covalently associated enzymes that catalyze two or more sequential steps in the metabolic pathway. Okay. So why such a complex set of enzymes? Because enzymatic reactions rates are limited by diffusion with shorter distance between subunits. Enzyme can almost direct the substrate from one subunit to another. Channeling metabolic intermediates between successive enzymes minimizes side reactions, and the reactions of a multi-enzyme complex can be coordinatedly controlled. Correct? So you see this is a covalent modification of uricylate pyruvate dehydrogenase. Okay, so it can be made active and inactive accordingly. The addition of a phosphate group. So these are the 5 reactions of the pyruvate dehydrogenase multi-enzyme complex. So this enzyme requires 5 coenzymes and 5 reactions. So these are the coenzymes and the prosthetic groups of pyruvate dehydrogenase. This is the domain structure of dihydrolypoyl acetylase E2. So in general, the TCA cycle is inhibited by a high energy charge and stimulated by low energy charge, right? The glycolysis produces pyruvate by oxidation of glucose, and the pyruvate is then oxidized to acetyl-CoA in the mitochondria. Acetyl units are oxidized to carbon dioxide by TCA cycle in the mitochondrial matrix, and the energy released during both the oxidation reactions are collected by NAD plus and FAD. So NADH and FADH2 carry energy in the form of electrons. Okay. So where do all the NADH and FADH2 go? So they all move ahead to the ETC or the electron transport chain. Okay. Moving on to the next question for today. Anaplerosis involves the formation of the vital intermediates of a particular pathway through other reactions, while cataplerosis involves the removal and subsequent transformation of certain intermediates to prevent their accumulation in the mitochondrial matrix. So which of the following intermediates are subject to anaplerosis or cataplerosis? So as we were discussing the previous question, these two terms came up. So we said that we'll discuss it in the next question. So this is the next question. So which of the following intermediates are subject to anaplerosis or cataplerosis? These are the four options: pyruvate, fumarate, succinyl-CoA, and alpha ketoglutarate. Which one of these do you think is the correct answer? Anybody? Okay, so we'll move ahead with the question. The answer will be succinyl CoA and alpha ketoglutarate. Now we'll discuss about these two terms. So in aerobic organisms, citric acid cycle is an amphibolic pathway that we discussed in the last question, one that serves in both catabolic and anabolic process, right? Since the citric acid does both synthesis, anabolic and breakdown, that is the catabolic activities, it is also called an amphibolic pathway. Now this is an amphibolic, that is it is both anabolic and catabolic in its function. Okay, both degradative and biosynthetic. Now, central metabolic pathway and amphibolic pathway is a set of reactions which permit the interconversion of several metabolites and represents the end of the catabolism and the beginning of anabolism. Correct? So it's a series of reactions that degrades acetyl CoA to yield carbon dioxide and energy, which is used to produce NADH plus HH plus and FADH. Now the Krebs cycle connects the catabolic pathways that begin with the digestion and degradation of foods in stages 1 and 2 with the oxidation of substrates in stage 3. This generates most of the energy for ATP synthesis. Now, the citric acid cycle is the final common pathway in the oxidation of fuel molecules. In stage 3 of metabolism, citric acid is the final common metabolic catabolic intermediate in the form of acetyl CoA. Now, this is why citric acid cycle is called a central metabolic pathway. Now, moving on to anaplerosis and cataplerosis. So, anaplerosis is a series of enzymatic reactions in which metabolic intermediates enter the citric acid cycle from the cytosol. Whereas cataplerosis is the opposite. It's a process where intermediates leave the citric acid cycle and enter the cytosol. So, in muscle, anaplerosis is important for increasing citric acid throughput during periods of exercise, and there is some evidence that anaplerosis is required for a glucose-induced rise in mitochondrial ADP production. So, Krebs cycle is amphibolic. It contains both anabolic and anabolic reactions. Okay. So the energy from oxidation of acetyl CoA is stored in reduced coenzymes, whereas several intermediates and precursors in the biosynthetic pathways. Now this is a diagram of the citric acid cycle indicating positions at which intermediates are drawn off for use in anabolic pathways. If you can see where with red arrows, and where anaplerotic periodic reactions replenish depleted cycle intermediate, those are dark blue arrows, dark green arrows, sorry. You can see these. Now, citric acid cycle intermediates are always in flux. That is, reactions that replace cycle like anabolic pathways are a set of metabolic reactions which replace central metabolic pathway. So you see, the citric acid cycle always it's in flux. So the reaction catalyzed by pyruvate carboxylase that replenishes oxaloacetate to the TCA cycle is a good example of an anaplerotic or filling up reaction, right? And this reaction assures that there is sufficient oxaloacetate for condensation with acetyl-CoA. In fact, acetyl-CoA stimulates the pyruvate carboxylase, and this reaction is most important, especially in liver and kidney. So this is an example of an anaplerotic reaction. So the anaplerotic or filling up reaction that connects pyruvate to the Krebs cycle. So these are all examples of anaplerotic reactions. If you see, and the exact organisms where and tissues where this happen. Now, other important anaplerotic reactions include the anaplerotic reactions that are sources of succinyl-CoA. So, succinyl-CoA is formed from fatty acids with an odd number of carbon atoms via propanyl-CoA. Succinyl-CoA is also formed from propanyl-CoA generated in the breakdown of the branched chain amino acids isoleucine, methionine, and valine. So oxidation of odd chain fatty acids leads to the production of succinyl-CoA from propyl-CoA. And transamination and deamination of amino acids leads to the production of alpha-ketoglutarate and oxaloacetate. Now these are the energy pathways in the body. You can see the dietary or the energy reserve sources in cells will be triglycerides, polysaccharides, proteins. Circulating energy substrates in the blood will be fatty acids, cholesterol, ketone bodies, glucose, lactic acid, amino acids, and the intermediates of the aerobic respiration in the cell. So this is, we have already seen this. The first step is the digestion and the hydrolysis. Then degradation and some oxidation to the smaller molecules, correct? And then oxidation to CO2, H2 and energy, which happens inside the mitochondria, the Krebs cycle. We have also talked about this interconversion of glycogen, fat and protein, conversion of glucose into glycogen and fat. This is also we have touched upon. So how fats break down and feed into the TCA cycle. Basically, what happens? Fatty acids and glycerol, they do beta oxidation, and coenzyme A might be produced, which can then feed into the Krebs cycle. So breaking down fat for energy produces acetyl-CoA, which feeds directly into the citric acid cycle. So both fatty acids from lipids and amino acids from proteins form acetyl-CoA, which enters the citric acid cycle for continuation of their degradation or catabolism. Okay, so the carbohydrate part is pretty easy, glucose, pyruvate, but amino acids and fatty acids like proteins and lipids also ultimately end up in the Krebs cycle. So proteins may serve as excellent sources of nutrient energy. Now catabolism of amino acids provides succinate, oxaloacetate, fumarate, alpha-ketoglutarate, pyruvate from glycolysis, and acetyl-CoA stimulates the pyruvate carboxylase. Proteins can also be broken down to feed into the citric acid cycle and generate energy. Under extreme starvation in animals or during senescence in plants, polypeptides are cleaved to amino acids. Amino group is cleaved of amino acids, forming an organic acid that can enter the citric acid cycle. So you can see this is the pathways by which amino acids can be catabolized. Okay. This is the entire schematic of all the pathways. Now, degradation of the amino acids that produce pyruvate or intermediates of the Krebs cycle are shown at the points at which they enter the cycle. Okay, so these are shown in the at the same points. This is the Krebs cycle schematic again. So, strategy followed by the cell in the catabolism of amino acids is to convert all 20 common amino acids into one of the following 7 compounds useful in gluconeogenesis or the citric acid cycle, like acetyl-CoA, acid-acetyl-CoA, pyruvate, oxaloacetate, fumarate, succinyl-CoA, alpha-ketoglutarate. So, most of the metabolic pathways of the body are channeled through these 7 molecules. Now, the great importance of the 2 slides above resides in the fact that they highlight some of the molecules that are the major players in the metabolism in the body. Okay, so these last 2 slides basically indicate the position of these molecules in the citric acid cycle and emphasize that the Krebs cycle is indeed a central metabolic pathway and an amphibolic pathway which has both a catabolic and anabolic role. Now, carbon atoms from degraded amino acids are converted to the intermediate citric acid cycle or other pathways, and glucogenic amino acids, which are shown in orange boxes, produce carbon skeletons that can form glucose, and ketogenic amino acids, shown in green boxes, can produce the ketone bodies. This schematic shows how the protein and the amino acid catabolites feed into the TCA cycle. So this schematic shows anapterosis involving amino acids entering the citric acid cycle from the cytosol. Okay, so we come to the end of the question and we come to the end of the session as well. So this was the last extra session, and we discussed 2 of the questions here. So if any one of you have any questions, we can discuss that now or we can conclude the session. Anybody? Okay. Since there are no questions, we can conclude this session. Thank you.